Hello, NE204, and welcome to Lecture 1 of Week 3. What we're going to discuss this week is observations of action potentials or spikes, and think about some real-world examples, and also develop some tools to characterize these spikes, including graphical representations and ways to summarize data that we'll eventually apply in MATLAB on Friday. So let's start by considering an experimental observation. Here we have a rat brain, and a very talented electrophysiologist is able to remove a thin slice of a region of this brain, the hippocampus, which is shown in this figure in light blue. There's a thin slice of hippocampus. That slice of hippocampus is placed in a petri dish, and then a single neuron is patched. So imagine we have this slice of brain tissue. It's been cut out of a living rat and then placed in a petri dish where it can be preserved and studied. All right, so what does it mean to patch a neuron? Well, let's first consider a recording setup. So here is a graduate student or former graduate student from BU. He's set up this electrophysiological rig where there's a brain slice. It's highlighted here under this microscope, shown here in green. There's a small electrode that's inserted into that slice of brain tissue, which you could just barely see here as a device to control the electrode. And then there's a microscope that's used to guide the electrode so that the individual neuron can be observed. So this is a typical experimental setup that you might see here at BU or at another lab. And what does it mean to actually patch a neuron? So we discussed this very briefly earlier in the course, but here's the idea. The idea is to stick a sharp glass electrode onto the surface of the cell and rupture the membrane. And that's illustrated graphically here, where there's a sharp electrode. It's put right up to the surface of a neuron. Here's the soma of a neuron. So this electrode is put very close to the surface. And then that surface is ruptured, so you get a flow of ions that can move in and out between the electrode and the neuron. As an example of that, here is an actual image of an electrode patching a cell. And if you look very closely here, you can see the hint, this ghost of an electrode shown in blue, that's patching an individual neuron. So you can see a bunch of neurons here, and there's one neuron that this electrode is targeting, the patched neuron. Here's another example where if you look very closely at this figure, you'll be able to make out the individual electrode, which is shown here, this ghostly image. And then you can see a cell body. This is what an experimentalist, an electrophysiologist would see through a microscope. So trying to target this electrode to patch an individual neuron. So that's the experimental setup. A glass electrode is stuck into a petri dish, and an individual neuron is patched and recorded. And what's actually observed is the voltage activity of this single neuron. And what is actually found when this voltage activity is investigated? Well, it turns out there are lots of different types of activity that can be observed from an individual neuron. So here are just some example spike trains that we might see from an individual neuron. Here's some other examples and even more examples. And hopefully the details aren't necessarily important here, but the important point is that lots of different activity can be produced by individual neurons. So we get these many different types of spiking behavior that can be observed, again, from an individual neuron that's been patched in vitro in this small petri dish. And one other observation we can make is that this spiking activity is rather complicated. So it can have different features and shapes. And what we'd like is to develop tools that can tell us a bit about these features and shapes. So what we're going to do to start is we're going to try to simplify. We're going to ignore the detailed voltage dynamics. So why would we do this? Well, one answer is that the data is so complicated we'd like to simplify and make our analysis a little bit simpler. So let's consider, for example, this spike train, which is shown at the top of the figure. We can ask lots of questions like what affects the time between spikes, or what determines the shape of the action potentials or spikes. So those are all interesting questions we can investigate, but we're going to ignore those for now. We're not going to worry about the shape of an action potential for now. And instead what we're going to do is indicate the time of each spike with a symbol. Okay, so to do that, our first step will be to divide the time axis into many tiny subintervals. So here's our time axis, which I've drawn here as a horizontal line. And we're going to divide it up into lots of tiny subintervals, which I've indicated here. So the time axis divided up into many tiny subintervals. And we're going to make each subinterval so small that at most one spike occurs in a subinterval. So choose any subinterval you want. I've indicated one here with a red arrow. This interval has either 0 spikes or 1 spike in it. So each interval has at most one spike. It might have none, but it can have at most one spike. And if a spike occurs in a bin, we're going to label it. And here we can use tick marks. So for example, if a spike occurs in a particular bin, we'll put this bold vertical line in that bin. So that will indicate a spike. So in this simplified representation of spiking, we're going to convert our observed neuron voltage activity, which is very complicated and rich. We can ask lots of interesting questions about it. We're going to take this complicated voltage activity and represent it as a series of tick marks, as indicated here. So let's investigate these tick marks just very briefly. Every time you see a spike, what are we doing? Well, here's a spike in our voltage trace. We indicate it with a tick mark. Here's another spike. We indicate it with a tick mark. A third spike, and we indicate it with a tick mark, and so on. So we're taking this complicated voltage trace and representing it as this series of tick marks. Or we could choose an alternative symbol. We didn't have to use these tick marks. We can use zeros and ones, where a 0 indicates no spikes in a subinterval, and a 1 indicates we observe a single spike in a subinterval. So notice that both of these abstract representations contain the same information. The tick marks, as drawn here, or the zeros and ones, as drawn in the second line, these both indicate the intervals, these tiny subintervals where a spike occurs. Okay. So again, focusing on this simplified spiking representation. We started with this observed spiking activity, which is rather complicated. It's a plot here of voltage versus time recorded from a single neuron that maybe we or a talented electrophysiologist has patched. From this, we produced two abstract representations of spiking. One could be this series of tick marks, and the other one could be this series of zeros and ones. So we've gone from the complicated voltage data to this abstract representation. Why is this good? Well, it makes our lives a bit easier. The representation is a bit simpler. Right? In this case, in this abstract representation, we're only indicating the times when a spike occurs. Why is this bad? Well, we ignore some details. So questions like what's happening between the spikes and the voltage activity, that's completely lost to us. We're only indicating with these tick marks or zeros and ones when the spikes occur. We don't know anything about what happens between the spikes when we just have these zeros and ones. All we know is that a spike did not occur in this interval, but we don't know what the voltage is doing. Also, in this case, these tick marks just tell us a spike occurs. They don't tell us the shape of that action potential or the height of that action potential. All of that information has been thrown away. And a reasonable question we can ask is which abstract representation is better? So we might ask, do we want to use these tick marks or these zeros and ones? And we'll see that they both have advantages as we go along. Sometimes we'll want to use this 0-1 notation. Sometimes we'll want to use this tick mark notation. Usually we'll use the tick marks for graphical representations of a spike train. And we'll use these zeros and ones because those are easier to manipulate in MATLAB. A more general question we can ask is, what's the best way to represent the activity we observe? Maybe we should be studying this original voltage trace in all of its details. Or maybe instead we should be studying these abstract representations because we can get a better handle on it.